Good morning, everybody. I think it's time to start. It doesn't feel like everybody is here yet. Maybe some people think they already know enough about structural information, which is fine. So, what do you already know about protein structure determination, experimental methods? Crystallography. Right. Oh, you've been reading the chapter. Again. Yeah. NMR. Yeah. EM. Yeah. Small angle scattering, you mean? Yeah. Okay. Right. So that's sort of the summary of today's lecture, right? What? This is on. The light is on. It's not only annoying, it's right, okay. That's correct. Where was I? Oh yes. I'm completely off my track. That's really cool. Yeah, so the reason that we have this lecture in the course is basically to give you a feeling of where the data comes from. Because, I mean, you can do structural bioinformatics basically just out of the PDB. You don't have to actually go and measure your own structures. Right? That's the nice thing about databases. But not having any knowledge of where the data come from, that's dangerous, right? Because you don't know which part of the data to trust or how to work with it. So that's why we have this lecture in the course. And so we start with the basics because that's where we always start. And so the way we can, because getting information about protein structures in an experiment is not easy. So you need all sorts of cool tricks. And fortunately we have electromagnetic waves, and proteins respond in interesting ways to lots of different wavelengths in that spectrum. And the important ones are highlighted. So X-rays are actually on the high end in terms of energy and frequency, very short wavelengths, and that's what we use. And at the other end, we have radio frequency waves, which we use to measure nuclear magnetism. That's for the NMR. If we have time at the end, I'll tell you a little bit about, let's say, the regular spectroscopy in the infrared and the ultraviolet, which is also used for not quite structure determination, but you can get structural information out of proteins using those spectroscopic techniques as well. Okay. That's enough about this spectrum. So the I'm not sure if most interesting is the right word for it. I'm going to close the door because I don't know what they're doing next door. But the methods that give most detailed information are usually the most interesting. It depends a little bit what you need to know about the protein. And so the two methods that really give high resolution results are the crystallography and the NMR. Morning. And actually, I also put cryo-EM here in the bottom right corner because it doesn't quite get to atomic resolution yet, but it's getting very close. So, does anybody, except the chemist here at the front row, does anybody know roughly the size of a carbon atom? What scale are we thinking of? Angstroms? How many? 1, 2, 3, 4, 5? 2. It's. I think the radius is about, or the diameter is about 3 angstroms. Carbon atom. So anything of 3 angstroms resolution or below is sort of atomic. Real atomic is about 1.5 angstroms, because then you have some precision with which to determine where this carbon should actually be found. And but cryo-EM is getting to resolutions of about 4 angstroms. Not quite there, but it's amazingly close. Okay. So, and the really cool thing about cryo-EM is that it's actually microscopy. So you actually literally zoom in on a molecule. So you can't really zoom in on the molecule and then see the atoms. That's not how it works. But you can actually image multiple individual molecules and merge those images together to get higher resolution. Or let's say a compound image. So you would be interested. There's actually a very cool figure in the book that I haven't actually had time to put in the lecture yet, which does that for a membrane complex in a bacterium. But they have it imaged as it's actually infecting a human cell in the culture. Right? So, and then you take like 1,000. You find 1,000 of those complexes or so in your images, and you combine those, and then you can get to resolutions of 5 to 8 angstroms. That's really cool. The other 2 methods are more indirect. Crystallography, you make a crystal, and you look at the scattering of, or actually the diffraction of the X-rays, and then you have to calculate back the structure. And NMR looks at nuclear spins, and you can do all sorts of tricks with energy transfer between nuclei, and they can be dependent on angles and distances. So you can measure with some accuracy, some or sometimes a lot if you're lucky, angles and distances, and then you can from that model the structure. So that's quite indirect. It works well. You get very high resolution data. This is quite direct, but it's also not easy. Okay, so we'll is there a clock here? There's no clock here. Cool. I have a clock here. We'll start with X-ray. This is the main method. Not because it doesn't have any downsides, but because it's actually if you get your crystals, if you get your protein crystallized, you can nearly always get a structure. So, so it works well for most proteins, except for the ones that don't crystallize, which includes transmembrane proteins, which is about 2/3 of all proteins. So, but NMR has its own downsides, and electron microscopy is still very, very labor intensive. Most of the structure that we have nowadays, by far, I think it's must be something like 85%, if it's not higher, the structures in the PDB, they come from X-ray diffraction. Okay, so that's why we start there. So this is sort of a very simplified view of the workflow for getting from a protein to a structure. There's a couple of steps. First of all, you need to have a purified protein. This is sometimes already a bottleneck because you need enough of the protein and you need to have it quite pure. Because if it's not pure, it won't crystallize. So that's one bottleneck. Then if you have a crystal, you get a diffraction pattern. That's no problem. But the crystal has to be good enough. Then from we'll get into the details in a minute. From the diffraction pattern, you can get the electron density, and you have a problem with the phases that I'll explain in a few slides. And then from the electron density, you can actually get the atomic coordinates, right? So the most important thing to remember, if that's the only thing you remember of crystallography from this lecture today, remember this. The 3D structure, so the atomic coordinates that are deposited in the PDB are not experimental data. They are derived data. The only experimental data that you have in your crystallography experiment is the diffraction pattern. Yes? So you cannot look at a diffraction pattern and say, I know where this carbon is going to be. It's very, so it's the electron density is closer because each of these blobs is going to be an atom, or maybe two atoms, or maybe an atom and a half, because the other half of the atom is not quite well visible. Yeah? But this is the data that we typically work with. It's not experimental data. That's important too. Okay. So now, what do protein crystals look like? Well, under a microscope, they look like this. If you're lucky, they're like 2-3 millimeters big. The bigger the better, because you get more signal out of them. But inside, how do you build a crystal out of proteins? Packing and stacking? Yeah. So the definition of a crystal is like a regular array of molecules. Right? So the typical example is salt. Here you have sodium and chloride, and then you have this nice regular packing of the two ions together. Sugar also crystallizes, you know that. Yeah? Those are small molecules. So it gets harder the bigger the molecules are, so it's not very easy for proteins, but the principle is the same. And these are a few views of different protein crystals. An important thing, so if you take a salt crystal, it's just sodium and chloride. That's nothing else. If there's water in it, it's no longer a salt crystal because it dissolves. That's different for protein, and it's the same for sugar crystals. It's different for protein crystals because protein molecules are quite big. There's a lot of space in between. Yeah, if you have a box of marbles, knickers in Dutch, there's a lot of air in between. So if you imagine the marbles being proteins, then instead of air you'll have water. So protein crystals are actually wet crystals. And so most of the contact points between the proteins are actually relatively small. Think again of the box of marbles. Proteins are not like marbles, but they're sort of spherical. And there's only like, if you have two spheres touching, there's only like a relatively small area where they actually touch. Right? It might be touching on a few places around the different areas, right? But it's not that it's not like blocks that are packed together and they're touching on all sides together, right? So most of the protein in the crystal is actually still interacting with water, not with other proteins. Right. So this also means that in many cases, if you have an enzyme in a crystal, you can add the ligand, and it might be slower than a solution, but it could still be catalytic. If you need cofactors or other stuff, you have to add them too, obviously. But many crystals are shown to have actually still enzyme turnover in the crystal. That's very nice, because that also tells us that the crystal is at least close to the native state, close enough that it's still functional. Right? Because one of the worries that you should have when you stick your protein in a crystal is that it's different from the environment in the cell, and the protein might respond to that by changing its shape. It usually does, right? And it doesn't. It's not, but it's not always that bad. It's the take-home message. And even if you don't have catalytic activity, you can still often get the ligand to bind. So that's also already quite interesting for two reasons. One, that still means that the conformation of the protein is still similar to what it should be. Morning, ladies. The other thing is that you can then actually make a structure with the ligand bound in it. And if you're doing medicinal chemistry, you want to develop the new molecule to inhibit this enzyme or to activate this receptor or whatever. That makes a lot of sense. Yeah? You can actually study the protein-ligand interactions quite directly. So, and if you're lucky, this is done routinely in pharmaceutical companies. You can make a crystal with a low affinity ligand bound in it. Then you try to wash it away, which doesn't really work. But then you add a higher affinity ligand, which will probably replace the previous one. And every time you put the crystal again back into the X-ray machine, and you record another crystal structure. So you can put one protein crystal, and then you can record the interaction with like a whole set of different ligands. Okay, so some more examples of crystal packing, a little bit more detail. And do you know what these, what's the word for that? Oh, sorry, first question, yeah.
So how long will the crystals usually, how long will the protein maintain active when it crystallizes it?
How long will it maintain? Well, that's a good question, I don't know. I think if you store your crystal carefully, there's no reason for the protein to fall apart. Why would it?
Say you look at freeze it like a big batch and I
Yeah.
can keep on using it.
Well, I don't know if you can freeze a crystal. I think that will probably, because then you crystallize the water. I'm pretty sure, or, well actually they do crystallography at low temperatures, so that must actually work. Good question. Oh, I know how to do it. They freeze it very quickly, so that water actually does not crystallize. That's actually, yeah. So, and then if you store it at liquid nitrogen temperature or so, probably even -80, I think it stays well indefinitely. I mean, there's not much that can actually access the protein in the crystal. So, right. So, what do you know what these? I can't think of the word. Boxes, thank you. Yes, that's good. I'm looking for a different word, doesn't matter. What these boxes actually represent? The repetitive units. Yeah, it's not on the side. Okay. Yeah, so why is that important?
The fraction will tell you the structure of the whole box, not only of one unit, not only of one item in the box.
Yeah, but it's actually the thing that builds up the crystal, right?
Yeah.
It's the repetitive. So if you don't, I mean, it's so this is actually. But so the thing is that you're actually measuring. Let's say that. I have to explain it going back from what you said, right? So you have. I have to think about what to do in which order. Okay. No, I'll do that later. First, so this is just how the crystal is built up. Yeah, because if it's not a repetitive unit, it's not a crystal. We'll come back to how you actually then collect the data from the crystal. And so you can actually see that this is repetitive, right? So it's the same thing all over again. And this goes on from the molecular point of view infinitely. Yeah, I mean, obviously there's a size to the crystal, but in terms of the number of molecules that you have to draw on the board, it's relatively infinite. So this is very close to like stacked marbles, yeah. Although it would be hard to get marbles to stack with this nice hole in between. I think. Oh, I've tried this once. It is hard. But this one does something really different. It's more sort of like filamentous stacking, right? And here you actually have a lot of water in the crystal, obviously. It's about 90% water, so that's quite solid crystal. Yeah.
Is a crystal just like one layer of protein, or should I?
No, this is three-dimensional. Yeah, it goes in. Yeah, but it's hard to draw on the board. So the actual, yes, but it's a good point. So the actual repetitive unit is not like this flat shape. It's actually a box. Right? So, do I have a figure of that? No?
The last slide, basically.
Yeah, the previous slide. Yeah, so this is more or less the three-dimensional view, right? But it's not good. I'm gonna look for a nice picture. Okay. So this is what crystals look like, right? So now let's go back to our working diagram and look a bit at the first step of the workflow, which is getting your crystal. So what would be problematic issues with getting your crystal? Besides producing the protein in enough quantities and purifying it, because that's also important, but let's suppose you already have a sufficient quantity of purified protein, Why would some proteins not crystallize easily and others do? You had a question or a suggestion?
Suggestion.
Yeah?
There's certain balance between what's kind of solved, etc. Together.
Okay, so the conditions, right? So, yeah, so this is one of the magic ingredients in crystallization. Salt concentration, different kinds of co-solvents, the pH, temperature. There's kits you can buy that actually help you screen like thousands of different conditions to find the exact right one that will get your protein to crystallize. So that's one part of it. But I was thinking more of properties of the protein itself, because some proteins just crystallize quite easily, and other types of proteins are very hard to crystallize. Any ideas? Yeah?
For example, if they expose hydroatomic stretches, they actually precipitate.
Yes. Yeah. So precipitation is a bit like crystallization, except it's not regular. You get an irregular clump of protein. Technically, you'll get a diffraction pattern out of that, but you need the regularity to be able to calculate back the positions of the atoms. So that won't be very useful. Yeah. So if it aggregates, it's done. Yeah.
I guess the molecule can have different conformations, so you get irregularity from the protein.
So floppy bits, to rephrase that, in the protein will actually. So there's actually two problems to that. One is that it might not fit in the crystal in the same way all the time, right? So you might get different conformations inside a crystal. That will be. If it's only two conformations and it still gets nice crystal packing, you could imagine that you have this repetitive unit, and there's a different conformation for this copy of the protein, and then for that one, and you repeat that. That's fine. That sometimes happens. The other problem with floppy bits in your protein is an entropic cost. Because if your protein is in solution, everything can move around. In the crystal, this is much more restricted. So it might not actually be entropically or thermodynamically favorable for the protein to form a crystal, because the entropy is, the lost entropy is too high. Yeah. We'll get back to thermodynamics next week. We mentioned it already before, right? Okay. So, and this particularly if you have multiple domains with flexible linkers, then it just probably won't fit into a crystal. Termini of proteins are also notoriously floppy. They could be like a binding unit that's flexible, but it becomes a helix when it's bound to whatever it needs to bind to. But if you don't have that whatever it's binding to in your crystal, then it will still be floppy. Going? Yeah. Okay. So there's an easy solution to this. How would you solve this?
Truncating the protein?
Yeah, just cut it off. Yeah? And this is routinely done. So that's also why if you compare a PDB structure with the UniProt entry, you'll find that in most cases the PDB entry has fewer residues than the sequence of the protein coming off the G. Because, well, sometimes it's post-translational modification, but usually it's just because it's been truncated by the crystallographers, because they were crystallized with. Yeah? And then you need a few trials, cut off 10, then 20, then 30 residues. If 30 doesn't work, you cut off 100. And then if that works, then you do 50, and then you sort of optimize it. You want to cut off as little as possible, but you still want to have a crystal. How will you do that for the multi-domain protein? Do it multiple times. Yeah, so you cut the different domains. How do you know where to cut? If you have a structure, it's fairly easy. It's not trivial. Fairly easy to find where the domains are. That's for a later lecture. Or maybe did Jaap talk about domain prediction in sequence analysis? You can do this from domains. This is for a different lecture, but you can actually recognize domains by looking at sequence, which is convenient because the reason that you're doing crystallography is probably because you want to have a structure for this protein, and you don't have it yet. Yeah. Okay. If you don't know, you could just that's what people sometimes do. You just cut it randomly. You tell, I think it's probably 3 domains. So you have 700 residues, you cut it into 3 equal pieces, and then you see what happens. Not very efficient, but if it's the only thing you have, it's working. Okay. The other sort of broad category is membrane proteins. And they have 2 problems. One is the problem that you already mentioned, that they have, because they need to bind to the membrane, they'll have big hydrophobic patches that interact with the lipid. It depends a little bit on what kind of membrane protein you have, but usually they have this to actually interact with the internal hydrophobic part of the membrane. So if you take them out of the membrane, then they'll aggregate. Yeah, that's one problem. The other problem is that usually the structure of these proteins is only stable when it's in the membrane. So if you take it out of the membrane, you get a different structure, or it's not stable at all, so it won't even crystallize. So the trick is to actually have sort of a bit of membrane-like environment in your crystal, and still have it crystallized, and that's very tricky. So what's usually done is you add some kind of detergents, which are like lipids but a little bit smaller, and then you get sort of a blob of like half a micelle around the membrane area of your protein, and then it should still crystallize. But the big problem with that is that these lipids, they're like the flexible parts of the protein. They will be irregular, and so they might not actually want to. They actually help keep your protein in solution as well, instead of crystallizing it. So there's a delicate balance. So that's the reason why, although membrane proteins are about 60% of most genomes, they are less than 15% of the PDB. Fortunately, cryo-EM is very, very good at membrane proteins. So we're getting somewhere, but it's going slow. The other part is, well, I say glycoproteins here. This is the major class, but it should actually be post-translational modifications, because there's lots of things that are being done too on or at the protein after it's been synthesized on the ribosome that could be very important for its functional role, and particularly insect mammalian cells, so higher eukaryotes. They have a lot of this post-translation modifications. And first of all, if you don't have them, you might actually have a different structure of your protein, or it might not be stable. Second, because they're not encoded in the genome, they will typically be variable. So they have like a crystal with 100 million copies of your protein, but each of them has like a unique variation of post-translation modifications that might not even crystallize, but if it does, you'll have a mix of signals, and you have to sort out. So the most problematic class is glycoproteins. So you have these glycans, so that's sugars, and it can be anything from one sugar molecule to a whole polymer of thousands of sugar entities. And they have 3 problems. They're heterogeneous because they're enzymatically attached. They will have different shapes. They're floppy, so they don't want to crystallize, and they're sugars. They're hydrophilic. They want to stay in solution with the water. Yeah? So sugar only crystallizes without water. Right? Proteins can only crystallize with water. So this doesn't work. You can't crystallize. As far as I know, you can't crystallize. Maybe somebody's come up with a very, very cool trick to do it, but as far as I know, people can't crystallize glycosylated proteins. Okay. So, where are we? A little bit about the experiment. So this is a very schematic view again. We have the crystal over here in the middle. It's mounted on let's say a bracket. Can be lots of different things. It's usually like a small loop where the crystal is sort of wedged into. There's liquid nitrogen to keep it cool. Different reasons. One of them is because you're going to shoot X-ray onto your crystal, which typically is energetic, so it will actually heat up your crystal. You want to get rid of that heat before your proteins and your water starts evaporating. The other thing is that if you want to have high resolution structure of your protein, you need to dampen the atomic motions. If you don't go to liquid nitrogen temperature, like if you do it at room temperature, you're stuck at about 3.5 angstrom resolution because the atoms are moving too much. The fourth thing is the goniometer, because you need to actually have I'd say that exposure of your crystal from different angles, but you need to know exactly which angles they work. So you rotate it in different directions. That's a very cool apparatus that does that reproducibly. And finally, you have the detector over here. That used to be a photographic plate, but nowadays it's a very high resolution CCD camera. And the final thing is a very simple it's a slab of metal. It's called a beam stop. Because not all the X-ray radiation is diffracted. Actually 90% or so of it just goes straight through the crystal. And you don't want it. You don't want that. Right? So it's just absorbed by this metal. And then you can see the diffraction pattern. It's like when you want to observe the corona of the sun, it's best done during a solar eclipse because the moon is in between. So you block the direct radiation and then you see the output. Okay, now I have a problem here because OpenOffice sometimes loses an embedded picture. There was supposed to be, I only just noticed, that there was supposed to be a diffraction pattern. So I'm gonna quickly pick up last year's lecture and and show you the appropriate sound. Good. Yes. Yeah. There was okay. So this is just a random diffraction image inherited from teacher Sixma. She's doing crystallography at the Dutch Master Institute. So here you see the white bit is where there's no radiation hitting the detector because that's where the beam stop was. Then the black dots, can you actually see them? Should I turn down the lights? So the black dots are actually the diffraction, the diffractions that we're interested in. And you can see they're sort of on a lattice, right? So you can see these lines, it's like a hexagonal lattice, but you see intense points in some of the lattice points, and you see nothing in some other areas. That's actually where the information is in. Yeah, so it's not quite about the location of the points, but it's about the different intensities at different locations. And the low resolution information is at the low angle, so that's close to the center. And if you go out to further distances, that's where the let's say the high frequency information is. So that actually gives you the information about the small spatial scales. So that means high resolution information for your atomic locations. So an experienced crystallographer, when he sees the first diffraction image coming out of the experiment, can immediately see whether it's going to be 8 angstroms, so basically useless, or 3 angstroms, which is cool, or 1 angstrom, which is like really very good. Yeah? Because they know, I don't know exactly, but they know that, this spot there, that's 2 angstroms. So pretty okay. Okay. Then there is a bit of diffuse scattering. This ring here is the scattering by the water molecules. I told you so. It's actually frozen very quickly. So the water is amorphous, not crystalline like we have with ice. But it does scatter, so you get this ring of diffuse scattering. And you get also some scattering from the loop where the crystal is mounted. Those are not so interesting, but if they sort of are too bright, then you lose information that you actually need. And then the last thing, so these refractions are actually on letters, and they're enumerated with 3 indices. And 2 indices make sense, right? Because you have a 2-dimensional plane. But I told you we're gonna actually rotate the crystal, so you should actually think of this as being a 3-dimensional diffraction pattern. That's why you need 3 indices. Yeah? And for each index point, you have an intensity, which can be 0 if there's no spot, or anything higher number if there's a spot. Okay. So now, how does this diffraction work? Why is there any information about atomic locations in this pattern of points? Waves? And then what? Anybody know Bragg's law? It's not about who's best at something or who thinks they're better at something than somebody else. It's not about bragging. Bragg's law? Yeah? Yeah? Nobody? Okay. This is Bragg's law. So look at the part over here, and you have radiation coming in. It's labeled primary beam here, and it's drawn like straight arrows, but you should imagine that this is a wave light propagating, and it refracts on this point, which is probably then an atom, or to be more precise, it's the electron cloud around one of the nuclei, and then another part of the incoming beam is diffracting off another atom. Right? Now, the thing is that because there are different atoms, there's a difference in the length of the path that's been traveled. So if this radiation is synchronous, there's a different word for that. Coherent, meaning that all the waves travel in the same rhythm, then because of this path length, they'll get out of phase here. Yeah? Makes sense? Okay. So now, depending on the angle, you'll have a difference in how the different waves have changed after refraction. Yes? Okay, now you know what 2 waves do when they meet each other? Ever, yeah, you get the sum of both, right? Ever thrown 2 stones in a pond? Yeah? That's what you should think of. Okay, so, but then the thing is, now they can be either in phase, so if the sum is actually double, or they can be out of phase. This is not as easy as it looks. They can be out of phase, which means the sum is actually 0, because when one goes up, the other goes down. So that's called destructive interference. And the other one is constructive interference. And that means that, so this is too complicated now. That means that for some angles you'll get, you get them to add up, so you get a spot. And for a slightly different angle, you get them to actually cancel out. And this angle is very precise. If it's not precise if you only have 2 points. Right? So that's where the next slide comes in. These points are actually the individual atoms. Let's say one atom, let's say the C alpha of residue 3 in my protein. This is C alpha residue 3 in this copy of the protein. This is the same one in the other copy, and this and so on. This is only, I've only drawn 3 molecules here. There's millions of them. Right? So now you should imagine that you have millions of secondary beams coming off in a particular direction, and only in some directions do they all line up. If you get the directions slightly off, then they will be randomized and they add up to 0. So that's why you get these precise spots in some locations and nothing next to it, and then another spot a little bit further on. Yeah? Now that you can start to imagine how this sort of, at least, is sensitive to the locations of all these atoms. Yeah? And it's not just for this atom, obviously. You should draw a diagram like this for each of the 1,000 or so atoms in this protein molecule. And all that together makes up the position of these spots. Now what this adds up to is basically a Fourier transform. So if you have the locations of all these atoms, or to be more precise, the density of all the electrons, but electrons are more dense around an atom, obviously, and less for different places. You get a Fourier transform of the of your electron cloud in your diffraction pattern. And we also have the mathematical operation of the inverse Fourier transform. So if we just reverse transform the diffraction pattern, we should get the electron density. Right? So mathematically this looks a bit like, oh, where are we going? This looks like this. Right? So this is the densities, rho, at x, y, and z coordinates. And this is the structure factor f, which is the intensity of a spot at this position, HKL, in the three-dimensional lattice that I've explained. And there should be an equal sign, I think. And so this is actually, there's two versions of this, but you should imagine that this is actually, in this, this is how most people write it down, but then the f is actually a, looking for the word, starts with a C. Yes, a complex number. If you write that out into two components, you get the actual f's that we've measured, and the alphas, which is another component. And they're called the phases. So in addition to knowing knowing how bright the spot is, you should also know what the phase of the wave was at that spot relative to the phase of the incoming radiation. Unfortunately, in crystallography, with this X-ray radiation, we cannot measure that. So we have only this part of the information and not that part. So if we would have that, it's easy, you do the reverse transform, you have your electron densities, you're done. If you don't. Okay, so now is this a really big problem because it's only half of the information, and I'll just show you that it is. So you can actually, so it's a Fourier transform, so you can do that on any three-dimensional data, but you can also do it on two-dimensional data, you can actually do it on one-dimensional data as well. I show you an example here where it's done on two-dimensional data, and these are Karl and Hauptmann, two pioneers in crystallography. So we're talking 1950s, 1960s. I'm pretty sure that I haven't checked that they also both won Nobel Prize, probably in chemistry. Okay, so now we can calculate the phase from doing the forward Fourier transform from the densities. We can calculate the phases and the phases and the amplitudes for Karl, and we can calculate the phases and the amplitudes for Hauptmann. And then we do the reverse Fourier transform using Karl's amplitudes and Hauptmann's phases, and Hauptmann's amplitudes and Karl's phases. And now if you look carefully, you can see that Karl now has Hauptmann's glasses and bald head. And his hair, and Hauptmann got Karl's bald head and lost his glasses. Yes? So there's a lot of information in these phases that we actually need to have to be able to do this. So this is just to illustrate that this is really a big problem. Now there are a number of solutions for that. It's not so important to have the details. If you have extremely high resolution data, low distances, small distances, then you can sometimes just infer the phases from the amplitudes. There are some tricks where you actually you replace some atoms in your protein with a heavy atom, and then this gives you shift in the amplitudes. And from the shift in the amplitudes, you can calculate the phases, or at least estimate the phases to some respect. It's called isomorphous replacements. There's some other tricks you can do. You can use different wavelengths, because if you do the experiment again with a different wavelength of X-ray radiation, from the difference in the diffraction pattern, you can also estimate the phases. But this is all quite laborious. This is not really doable for proteins because we can't get this data so precise. This is very laborious in the experiments and expensive, because you actually have to make proteins where, for example, you have not a sulfur in your methionines, but a selenium or an even heavier atom. So you need to grow E. coli on selenium, methionine, medium, and so on. Really expensive. What is most often done for proteins is called molecular replacement, where if you have a structure of a homologue of your protein, you actually do the forward Fourier transform based on the homologue, and that's an estimate of your phases. Then you add your intensities for the protein that you measured, do the reverse transform, you get a first estimate of your densities. Then you do the forward Fourier transform again, you get a better estimate of your phases. You throw away the forward calculated amplitudes, you put in your experimental amplitudes again, and then you iterate it a couple of times. Okay, so this is, we'll get back to that in a minute. So, what's next? So if you've actually done this, then you get what I already mentioned, the electron densities. And that looks a little bit like this. And actually, there's an increasing number of density maps also deposited in the, I'm not sure if they go in the PDB or they go into an associated database, but at least you can access them also through the PDB. And it looks a little bit like this, right? So, there's a, oh, I went back to, I'm still on last year's. Oh, I fixed it. What am I doing? Oh, I fixed it here. Didn't fix it here. There's a symbol missing. Okay, so when you have these electron densities, you first have to start looking for your secondary structure elements. So you look for sort of threads or wires of density, and this is probably the backbone, and then the things that are branching off are the side chains. So it's all. So then you actually, if your resolution isn't very high, you actually need to know the sequence of your structure, because otherwise you don't have enough information. Because you can't. There's nothing in the experiment that tells you that this has to be a phenylalanine. You have to recognize it from the shape, and then you don't know necessarily which phenylalanine it is. Only if you can trace it back to the start, you can actually say, oh, this must be phenylalanine 356, not phenylalanine 211. Yeah? So you need to know the sequence. There are examples where a protein was actually threaded in reverse through the density. This is called threading. There's a different kind of threading that Sanna will talk about when about comparing structures and homology searching, but this is also threading where you actually try to put your sequence into the density as well as possible. And so there's an example of a PDB structure, it's of course being withdrawn already like 30 years ago, but it was threaded in reverse through the densities. If you're resolute, so here it seems implausible, right, that you actually get it the wrong way around. But if your resolution isn't so high, then you might actually have cuts in your backbone, because it's just a bit missing. Maybe it was a little bit floppy or whatever. So you don't really know where it goes. If you cut it here, then you might think that the backbone goes on here. You might miss that this should be a phenylalanine. Also, if the resolution is lower, you typically first start missing out the densities of the side chains. So then it's very hard to see the difference between a phenylalanine and a, what's this over here? Oh, that's also phenylalanine. Or the difference between a phenylalanine and a aspartate. Okay, so just to show you that. So this is what a density map would look like if you have 1 angstrom resolution data. So this is very, very clear. So if you have data that clear, you don't need to know the sequence. You can actually read the sequence from it's still not trivial because this is only shows like 1 bit of peptide. And you should imagine that this is like there's atoms everywhere, right? But then it's possible. Weird. At 2 angstrom resolution, it's similar to the first picture that I showed you. You can still see sort of where the atoms are. You can see the shapes of the side chains. It's quite clear where the backbone is. Yeah, but you can't really pinpoint individual atoms. At 3 angstrom resolution, it's getting hard to distinguish different kinds of side chains. And this is still very clear data. Typically, as your resolution decreases, also the clarity of the data deteriorates. As I said, you might have cuts in the density where the backbone should have been. That makes it also very difficult to recognize bits of your structure. Then, at 4 angstrom resolution, it's getting hard to see what is backbone, what is side chain, and you might miss complete side chains and so on. You might start missing complete sections of backbone. So these densities that we're showing here, they're actually very clean because they were constructed. They were not measured. They were calculated based on the bit of molecule that is shown here, and therefore you also know exactly the phases. Right? So real densities are not that good. Okay. So after you have all of that, you go into what's called refinement, which I already sketched a few minutes ago. So if you have your first calculated densities, you calculate where your atoms are, and then from that you can actually make a better estimate of your phases. And then you can iterate this process a couple of times. The problem is there's no guarantee that this converges to the right solution. If you start with particularly bad guess for your phases for whatever reason, then it never gets better. You might get things horribly wrong. Yeah. So this also involves quite a lot of modeling in the sense that we know it. So known bond lengths, angles, and so on. Even force field calculations, energy minimizations, and sometimes even simulation methods are used to help this process along. So there's a lot of model knowledge going into this on top of the experimental data. And depending on the quality and the amount of experimental data that's there, it's more or less a mix of that. So ideally, you would like your model to just be a reflection of your experiment. So your final three-dimensional protein structure to be just a reflection of the measured experimental data. But as the data becomes less quality and less in quantity, it's going to be more of the model and less of the data that actually ends up in your PDB structure. Yeah. Okay. Finally, and this is actually starts already during the refinement process. There's a step that's called validation, and that tells you something about how good your model looks, your structure looks. And how would you know that? Well, you can actually compare it with known structures, right? So you look at things like side chain packing. So if you have a phenylalanine buried inside the protein, there's quite some typical contacts that it usually makes. So you can look at whether your phenylalanines in your new structure actually make similar contacts, or whether they're too close or too far away, or with the wrong type of atoms and so on. You could do similar things with your Ramachandran plot. If you have backbone angles that are literally off the chart, yeah, then you know that there's something wrong in your structure. So there's methods that actually do this automatically, and the basic idea, ingredient that they do is they've derived distributions of certain parameters from known structures, and then they look at the new structure and see whether it follows similar distributions. So this is a very powerful method. It has one drawback. What's the drawback?
There's a bias towards known structures.
Yes. Yeah, so if you're starting to make structures of, let's say, exotic membrane proteins, they might actually be very different in some respects from the known structures, which is dominantly soluble proteins, right? Doesn't have to be a lot different. So usually it still works okay, but there are some differences there. Yeah. Okay, so then before we go for a break, after the break we go to cryo-EM and NMR. The final thing is to mention the B factors. So they are actually sort of a residual of the match between your measured experimental data and your protein structure. So once you have a structure, as I already told you, you can do the forward Fourier transform. You get your structure factors and your phases. You can compare it with the measured structure factors, intensities, and so the deviation between those is actually a measure for the quality of your structure. And this is recorded at a global scale in what's called an R factor, just like the residual, and there's different variations of it. It's a very easy trick to get your R factor lower, is that you just add water molecules in your crystal, in your structure, not in the crystal, they're always already in your crystal. You add them in your structure, and then that's a very easy way to get the R factor lower. The other thing is the B factor values, because it's a similar kind of thing. It's calculated in a different way, but it does it by atom, and so for each atom it basically tells you how well the data restrains the position of your atom. So basically, if you shift this atom in different directions, does it actually make the fit with the data worse or not? And for some atoms it does, so they get a very low B factor, they're very well determined, and other atoms, they for some reason or other, they're not restrained by it. And usually so there could be two things, it could just be missing data, but usually it's either that this atom can be in different conformations, or it's really just floppy, so it's just moving around all the time. And then it just doesn't actually show up in the diffraction data. So then whether you change the position of the atom in your structure, it doesn't really make the fit with your data any better or any worse. Yeah, so that's B factors. Okay, so it's both a measure of the quality of the structure, and it says something about the dynamics of the protein, the local flexibility of the protein. Okay, with that I think we have time for 10 minutes break, and then I'll try to go through EM and NMR in the last half hour or so. Are there any questions so far? No? Yeah, sure. Baby's.
Densities met zeg maar bepaalde vorm van densities met structuur.